Hello, everyone, and welcome to today's webinar, Introduction to Wastewater Surveillance System for Domestic and Imported Pathogens, Korea Real Cases. I'm Claire Wu from Thermo Fisher Scientific, a Senior Regional Market Development Manager for Asia Pacific region in Genetic Testing Solutions team. Today's educational webinar is presented by Labroots and brought to you by Thermo Fisher Scientific. I encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. Your questions will be answered at the end of the presentation or responded via email after the event. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. Before we start the actual presentation, I would like to introduce our speaker, Dr. Chung. So Dr. Chung started his early work on HIV studies at Korea NIH. He was one of the key persons in Respiratory Virus Division in Korea CDC, establishing the SARS-CoV-2 diagnostic testing during the pandemic. He had set up a comprehensive wastewater monitoring system after being promoted to be in charge of Honam province in Korea at CDC. And he's currently the director at the Division of High-Risk Pathogens in Korea Disease Control and Prevention Agency, where he expands his expertise and successes on wastewater monitoring nationwide using real-time PCR, as well as other technologies, which will be mentioned during his talk today. Now, let's welcome Dr. Chung to share his great work on wastewater surveillance. Thank you.
Thank you very much for inviting me to this webinar. I'm truly honored to present the work we have undertaken in conducting pathogen surveillance in South Korea and initiating wastewater monitoring. It is a privilege to share our experience, and it has become evident to us that wastewater monitoring can yield valuable outcomes. Let's get started. I would like to introduce a wastewater monitoring system that I have been working on to the monitoring diagnostic and imported infectious disease. I will present the following agenda. First, I will introduce South Korea's infectious disease surveillance system, followed by an explanation of wastewater-based epidemiology. I will provide details about South Korea monitoring system for overseas infectious disease and discuss specific cases of COVID-19 in the WBE that I conducted in the Honam region. Furthermore, I will describe the process and details of WBE monitoring for overseas infectious disease at the representative airport in Honam region. This is an explanation of surveillance method for pathogens. A surveillance method can be distinguished based on the method of data collection, differentiation according to the selected population, and categorization based on the reporting method, such as sample surveys or complete surveys. Furthermore, a surveillance method can be differentiated based on their purpose. With these criteria in mind, I will briefly describe the surveillance approach employed by South Korea. In South Korea, the infectious disease surveillance system relies on a reporting system where healthcare professionals report a suspected infectious disease when patients with symptoms visit hospitals. The structure of the system requires patients to exhibit the symptoms and visit hospitals in order for an investigation to take place. As a result, symptomatic individuals or those who do not visit hospital cannot be included in the investigation. Moreover, due to the selection of samples for investigation, it is not possible to survey all healthcare facilities, comprehensively resulting in the inability to investigate all infectious diseases across the population. To overcome this limitation in infectious disease monitoring, wastewater surveillance is performed. It allows for early detection, cost saving, and does not require any personal information. Therefore, it becomes possible to identify infected individuals without symptoms. For cases of infectious disease imported from other countries, such as when traveling abroad and then returning home, monitoring is done to detect the infection with the pathogens upon entry into the country. In South Korea, there are quarantine stations at airport and port where incoming travelers are surveyed for symptoms, and those who exhibit the symptoms are isolated or subjected to the diagnostic test. However, it is impossible to detect those in the early stage of infection or those who are asymptomatic and do not show symptoms such as fever, respiratory symptoms, or rash. Therefore, monitoring pathogens through wastewater surveillance or air sampling can be a good way to confirm the introduction of infectious disease. This can help determine if infectious disease and pathogens are transmission within the local communities. Wastewater-based disease surveillance and monitoring refers to the system that detects pathogens associated with the infectious disease in wastewater. Generally, people infected with a pathogen excrete them in various forms, such as stool and urine, which then flow into the sewer system and eventually to wastewater treatment plant. By collecting wastewater samples from treatment plant and conducting genetic detection tests in laboratories, the presence of pathogens can be confirmed. Additionally, sequencing the genetic material can identify the types or variants or the pathogens, which can inform the public health system. Although there may be limitation, wastewater surveillance has the following advantages. It allows for early detection of disease outbreak and real-time monitoring of pathogen concentration. Additionally, it enables analysis of pathogens at a group level rather than individual detection. Since wastewater is used as a sample, there is no invasive action on the human body, and resulting in cost saving. Early detection of various pathogen occurrences can lead to early warnings, allow for public health intervention to be carried out accordingly. And then the Honam region is located in the southwest of South Korea, including the famous Jeju Island, as you may already know. I would like to introduce the wastewater surveillance results conducted in a city located at the southern end of the Honam region. First, wastewater was collected from 3 wastewater treatment plants, and nucleic acid was extracted from 140 microliter without any concentration processes. Real-time qPCR was performed using the extracted RNA to detect the SARS-CoV-2 gene as a representative. Here are the results obtained from June to October. The results are presented as CT values, and they were compared with the number of COVID-19 cases in this region. The points where the number of cases peaked were marked in red, and the region with the lowest CT values were also highlighted in red. The regions marked in red with the lowest CT value indicated that they have the highest viral load, meaning that a higher amount of the viruses was detected in those areas. I have represented the previous result in a graph. The red dot represents the point where the highest number of reported cases were observed, as indicated by the red box. On the left, the blue boxes represented the region with the low CT values. This means that the early detection of increased viral shedding can be possible approximately 2 weeks before the peak in the number of cases. These findings are highly significant, and similar results can be observed when comparing them with the results from various other countries. Honam region CDC conducted a more detailed analysis on the wastewater samples previously collected by local province. I will explain the result obtained by applying a method that concentrates the sample using equipment, followed by nucleic extraction and simultaneously detect genes using the real-time qPCR. First, as most some people are already aware, the wastewater samples were concentrated using Dynabead after collecting 10 mL of wastewater. Sample concentration was achieved using enrichment bead. This process is compatible with both manual procedure and Kingfisher automation, taking approximately 1 hour. Strong anion exchange was employed in this enrichment step. The nucleic acid was then extracted from the concentrated bead. The extracted nucleic acid was analyzed using TAC on quantitative 12K platform. Concentration was performed 6 sewage samples per run. After the procedure, the elute from the same samples were combined, yielding 1060 microliter. The concentration process involved reducing sewage samples of 4 ml down to the elute 1600 microliter. This equated to 4 well, each accommodating 5 ml of samples for total of 2 samples. Nucleic acid extraction was performed on the elute obtained from the concentration process. The concentrated samples of 1600 microliter were further processed to yield a final elute volume range from 50 to 200 microliter, which could be adjusted based on the downstream analysis requirement. The advantage of the TAC method is ability to concurrently test a large number of pathogens. We utilize the TaqMan Array Card as a method capable of processing a large number of samples simultaneously. In collaboration with someone, we designed this approach to identify 60 different pathogens with specificity. So notably, it includes SARS-CoV along with respiratory pathogens such as adenovirus, rhinovirus, common coronaviruses, 229E, OC43, NL63, PIB124, as well as diarrhea disease-causing agents like adenovirus, rotavirus, norovirus, SAPO, and enteric bacterial EAEC, PC, SDEC. Additionally, we ventured into detecting the various blood-borne viruses and herpes virus, including pathogens responsible for the gastrointestinal disorders such as enteric adenoviruses. While the presence of these agents in wastewater remains uncertain, this endeavor marks our initial attempt in this direction. As mentioned earlier, here is a list of pathogens confirmed on the TAC. In addition to include the key pathogens, we also incorporated the PMMOV gene for wastewater validation to ensure the extraction of genes from samples and PCR amplification verification. We include genosynthetic materials and then Bacillus bacteria as reference controls. It has a significant advantage that allows designing and incorporating pathogen based on the characteristics of each region or specific interest. The procedure for performing the TAC is also very straightforward. After mixing the sample's nucleic acid for PCR with the reagent, a simple centrifugation step is all it takes to complete the process. As a method for detecting highly complex pathogens, it is truly revolutionary. After performing TAC, we obtain the amplification plot. These plots allow us to verify each reaction, and it's essential to observe if the actual application curve lines approximately. Of course, we can compile the result for each target pathogen separately and export them to Excel, which proves extremely beneficial for result interpretation and organization. The TAC used in this experiment was designed to detect bacterial, viral, and antibiotic-resistant genes as seen in the figure provided. In the analysis of the major observed pathogen from the result obtained using TAC, it was observed that the RNA of SARS-CoV-2 was gradually decreasing during the sample period, which aligned with the later part of the result shown in the previous slide. However, while the previous result indicate a complete downward curve based on the average CT values, we observed a slightly upward pattern in region A and a decreasing pattern in the other regions when analyzed individually. Furthermore, pathogenic E. coli was also detected in all regions, and particularly norovirus G2 was observed in all regions as well, this confirming that we were able to detect the genes of various pathogens simultaneously using TAC. Moreover, within the TAC itself, we were able to predict changes in quantity of viruses based on the obtained result. This study aimed to observe whether it is possible to detect specific pathogen in wastewater discharged from specific facility, rather than large-scale wastewater treatment plant, and use them as indicators of pathogen occurrence within those facilities. In the G region, there was a significant outbreak within 70 confirmed positive cases originating from a nursing home. It was a highly unusual case where the recombinant XAR variant of SARS-CoV-2 was detected. The reporting of patient cases preceded the investigation, and at that point we want to verify if wastewater surveillance was possible. Therefore, we conducted wastewater testing by identifying the CV line of nursing home to validate its utility for monitoring purposes. Two wastewater samples were collected from the nursing home based on the date June 12, 28 and then July 5th. It was assumed that the genetic materials detected in the wastewater originated from the patient in recovery phase. The assays labeled RENN represented real-time PCR targeting the specific SARS-CoV-2 genes. The number of next to them indicate that mutation kit used the observed SMP variation. Furthermore, in order to compare the variant of SARS-CoV-2 in community outbreak with those in the nursing home, wastewater samples from the local wastewater treatment plant were also tested for genetic detection. It was observed that the presence of SARS-CoV-2 genes in the wastewater from the nursing home exhibit the decreasing trend, while the wastewater from the community showed increasing trends in gene detection. The variant analysis was performed using some GeneXus, which is a highly suitable device for automatically generating sequencing results from low concentration of virus RNA in wastewater. The detected genes were further analyzed for variant. NGS was performed using some GeneXus, and the results were highly successful. Just like the confirm of the XAR variant in patients with high viral load, the same XAR variant was also identified in the wastewater from the nursing home. In the wastewater treatment plant of community, the prevalent variant during the period, namely the BA2.3 lineages, were identified. This was highly interesting as it demonstrates the ability to monitor pathogen occurrence in a single facility through wastewater surveillance. Additionally, the result confirmed the possibility of genomic surveillance using wastewater sample. The previous slide, which include the phylogenetic analysis, provided excellent results that confirm the specific outbreak occurring within a particular facility, distinguish it from the prevalent strains in community. This is a remarkable outcome, demonstrating the ability to identify cluster outbreaks within specific facility, distinct from the dominant circulating variants in the community. I will introduce the monitoring of pathogens in airport wastewater. In order to determine the possibility of monitoring imported infectious disease, including COVID-19, we aimed to perform pathogen testing by collecting wastewater from airport. The objective was to detect various pathogens with a particular focus on COVID-19. We utilized the TAC method to detect various pathogens, which we believed would be highly useful for wastewater surveillance. Among the various pathogens screened, we identified the major ones among the detected organisms. First, SARS-CoV-2 was detected in airport wastewater with positive finding from 2 different airports. Additionally, it was important that viruses such as adenovirus and bocavirus, which lacked an envelope, were predominantly detected, along with the detection of norovirus. However, one issue encountered was that although all the pathogens showed positive detection in red box, there are some instances where the graph did not display an apparently upward trend. It was important to carefully observe the graph visually, and we also recognized the need for analytic validation of the primers and provers used. The presence of PMMOV in green indicates the inclusion of human fecal matter in wastewater and could serve as a control group mainly detected in human stool samples. Quantification of viral load is crucial in wastewater surveillance system. Wastewater monitoring is conducted over an extended period to predict and monitor the occurrence of pathogen within the community. Considering this, digital PCR appear to be highly useful for accurately measuring the detection quantity of pathogens in wastewater. Thus, absolute quantification was performed on airport wastewater, yielding results similar to TAC and providing precise absolute quantification values. This digital PCR not only offers quantification but also provides the highest sensitivity and allows for accurate observation of PCR inhibition substance that may be present in wastewater through dilution. Therefore, it can be a valuable tool in wastewater surveillance. Genetic analysis of airport wastewater was also performed using GeneXus. despite the low viral virus concentration, highly useful sequence results were obtained. The analysis of whole genome showed very interesting result. I was very excited myself. VF5 variants, which are not usually reported in Korea, were detected in the second samples of J airport. This result of phylogenetic analysis. The blue box at that location is the VF5. At this point, the predominant strain in South Korea is distinctly identified as belong to the BA5.2 lineage. I searched in which areas BF5 is prevalent. At that point, the most reported place for BF5 was Japan. I was even more excited to see this result. The airport flight schedule and when wastewater samples are taken, as you can see, the wastewater was collected about 30 minutes after the Osaka flight entered the country, and it is estimated that there were COVID-19 positives along travelers in Osaka. At the point in the water, the quarantine failed to test positive. In other words, there is a possibility that a person infected with COVID-19 enters the country symptomatic or at an early stage of infection and shares the virus. But it is not confirmed because there are no symptoms. I conducted the phylogenetic analysis by downloading the genomic sequence of SARS-CoV-2 from samples collected in the Osaka region of Japan on and around the date of collection from the GIS-8 database. As expected, it was found to be very similar to the BF5 strain in the Osaka region. This confirms that it is possible to identify mutant strains of SARS-CoV-2 through wastewater, as well as estimate the route of transmission. In wastewater-based epidemiology, TAC is considered a very useful method. First, it allows for the simultaneously confirmation of a large number of pathogens at once. Additionally, it enables the observation of pathogen variation patterns over time and the assessment of whether human biomaterials is present in wastewater. While validation for sensitivity and specificity is necessary, utilizing already validated primers and probes can achieve very high sensitivity and specificity. Based on the experiment we conducted, TAC is suitable for screening tests in wastewater-based epidemiology. I think it is thanks to Genesis, a highly sensitive NGS, that genomic surveillance is possible in wastewater. Firstly, it doesn't require researchers with highly specialized skills because most of it is automated. Therefore, it's as easy as PCR, and even informatic analysis, including whole genome assembly, can be performed on the Genesis platform server without the need for a dedicated bioinformatics expert. Several advantages have been described, and it is hoped that it will be applied to various pathogens in the future. And also, nuclear extraction from wastewater was the most difficult part in the past, but I was able to experiment very comfortably using the Kingfisher system's automated equipment. Various concerns were presented. WBE through occurred. Planning is currently starting, and I think good results will be obtained. I will explain the result of monitoring infectious disease using wastewater-based epidemiology. Of course, what's crucial is a careful consideration of where to collect wastewater, the purpose of the collection, and how the analysis will be conducted. And also standardizing the method for detecting pathogens in wastewater is a significant concern for me. After predicting the occurrence of COVID-19, we need to think about what other infectious diseases should be added in the future. Such consideration require comprehensive planning in the context of One Health, which encompasses monitoring infectious disease across the human-animal environment interface. I will conclude my presentation. Thank you all for your attention.
Thank you very much, Dr. Chung, for your great talk. We will now start the live Q&A session of the webinar. Once again, if you have a question you would like to ask, please do so by clicking the Ask a Question box located at the far left of your screen. We will try to answer as many of your questions as we have time for. While I'm seeing questions coming in and our audience may still be submitting the questions, I would like to introduce our moderator for the Q&A session today, Hui-Su Yu. She is our Senior Technical Sales Specialist for Genetic Testing Solutions from Thermo Fisher in Korea. Hi, Hui-Su. Over to you to help reading out the questions received from our audience today to our speaker, Dr. Chung. Thank you.
Thank you, Claire. Thank you, Dr. Chung, for your presentation today. We did have a number of questions come in, so I'm gonna try to ask you these questions and some sort of meaningful orders, so we are not jumping back and forth too much. Yeah, first one I see is about TaqMan Array Card. In the slide number 15, you utilize the TaqMan Array Card to screen a lot of pathogens from wastewater. How do you convince the government or policymaker to adopt TaqMan Array Card?
Okay, thank you. Very good questions. In countries where wastewater surveillance systems were initially implemented, a wide range of pathogens was already being monitored. Among them, the announcement that SARS-CoV-2 could be detected was made. In my opinion, there was information suggesting that various pathogens were being detected in wastewater, and among them, there were concerns about various pathogens that could be monitored for their occurrence. Some of these pathogens could be directly discharged into wastewater and potentially serve as source of infection for humans. Given these considerations, I believe that the application of a method capable of simultaneously detecting various pathogens, such as the TaqMan Array Card, would be very useful. I had previously received information from Thermo about their GI panel, since there are infectious diseases with symptoms but unclear causes that require screening for various pathogens. I considered applying the respiratory panel to wastewater surveillance. This approach had proven successful in my previous research, resulting in a very promising paper. Therefore, I believe it would also be highly suitable for wastewater surveillance.
Oh, it's great to hear that we are being part of the national project and we will continue to support. Can you talk a little bit about what will be the plan for TaqMan Array Card adoptions in future across other public health and environmental research institutes?
Yeah, some public health and environmental research institute facilities in South Korea are already using TaqMan Array Card. They are well aware of the useful of TAC in wastewater surveillance system. However, budget preparation is a top priority when it comes to purchasing and operating the equipment. In the future, many public health and environmental research institutes in various regions are interested in using TAC for screening, not only for SARS-CoV-2 but also for influenza virus, norovirus, and antibiotic-resistant genes. TAC stands out as a powerful method for simultaneously screening various pathogens and resistant genes, which has garnered significant interest among many health and environment facilities.
Oh, great, thank you. We hope all the HNE will adopt TaqMan Array Card in the future. Next question says, what are the key challenges you faced when implementing TaqMan Array Card? How the team overcome it?
Yeah, it's a significant advantage to detect a wide range of pathogens simultaneously in large quantities. It's crucial to confirm the sensitivity and specificity for each pathogen for the method to be recognized as accurate. Therefore, even when using primers proofs for 40 to 60 different pathogens, it's challenging to validate the sensitivity and specificity for each one. It may not be feasible to do them all at once. Instead, prioritized ranking of target pathogens should be established, and validation should be proceeded accordingly. As a result, validation is essential, and ongoing discussions with Summer will be necessary as we continue to fine-tune the approach.
Okay, sure. We will definitely support your project. Okay, let's see next questions. How has the use of TaqMan Array Card enhanced the detections and monitoring of pathogens in wastewater surveillance?
Yeah, as you may already know, pathogens in wastewater are often present in very low concentration due to the dilution. Therefore, they are often challenging to detect using conventional genetic assays, resulting in many cases where no detection occurred. To address this, we have incorporated the pre-amplification step. Following the suggestion from Summer, we performed 16 cycles of pre-amplification before conducting TAC reaction. This pre-amplification process has been shown to increase sensitivity by over 1,000 times. In wastewater, achieving high sensitivity in detection is our primary goal, and we have made effort to detect all existing genes using this highly sensitive approach.
Oh, it's fantastic. Pre-amplification works well and able to increase the sensitivity in detections. Okay, thank you. And next question is, can you speak about, are there notable findings or insights that have emerged from the pilot data?
Yeah, firstly, we needed to confirm whether pathogen genes are indeed present in wastewater. While this fact had been mentioned in the literature, it hadn't been confirmed, and verification was necessary for our study. However, we had confidence in the use of TAC because we already knew it worked well for detecting respiratory viruses. Thus, we performed TAC reactions using wastewater samples that were known to contain actual pathogens, and we obtained consistent results. This confirmed our ability to detect various pathogens simultaneously in wastewater, including not only samples from patients with high viral load, but also wastewater samples. Additionally, we observed that CT values are reflective of changes in viral quantity, considering the presence of diverse pathogens in samples like wastewater. We believe that the method of simultaneously detecting genes in such samples would be excellent. Yeah.
Thanks for your confidence in our TaqMan Array Card. We hope you could share with us in future seminar for verification study result. Yeah. Would you like to take this? What will be your advice to other countries or government bodies looking to establish wastewater surveillance and utilize the TaqMan Array Card?
Yeah. As mentioned earlier several times, wastewater surveillance involves complex mixtures containing a wide range of pathogens. While there may be cases where you want to detect only one specific pathogen in such a mixture, conducting wastewater-based epidemiology often leads to our desire to detect numerous pathogens. Initially, we may have wanted to detect only SARS-CoV-2, but over time, the list of desired pathogens and genes can expand it to include influenza virus, norovirus, and then various bacteria, including those with antibiotic-resistant genes like E. coli and more. In this context, be able to simultaneously detect various genes using the existing qPCR primers and probers would be an incredibly useful method. While not all pathogens may be detected in wastewater, conducting diverse screening and subsequently performing a quantitative or confirmatory experiment can be highly valuable in monitoring occurrence and predicting changes in wastewater-based epidemiology.
Awesome. Okay. Yeah, I want to get one more questions. You mentioned in the slide number 26, you found SARS-CoV-2 variant from wastewater using GeneXus system. Is GeneXus more advantageous for wastewater surveillance than other NGS platform?
Yeah, pathogens in wastewater exist at very low concentration, often making gene detection challenging even with the PCR method. As mentioned in our genomic surveillance work, we were able to obtain whole genomes with about 95% coverage by placing very low concentration of SARS-CoV-2 RNA into the GeneXus system and performing whole genome sequencing. This demonstrates that for analyzing genomes in samples with very low concentration, including wastewater, NGS equipment like GeneXus is excellent in terms of sensitivity.
Well, perfect. Yeah. I still see a few questions not being answered yet, but due to the time constraints, we shall reply the questions via email. So I just want to say thank you again to Dr. Chung for your time today and sharing your important research. Yeah. Let me hand over to Claire again.
Yeah.
Thank you.
Thank you, Huisu. Thank you very much again, Dr. Zhong, for your great presentation and also the responses to the questions. I know we still have a few questions that haven't been answered. And as Huisu mentioned, those questions that we did not have time to respond today, or those submitted during the on-demand period will be addressed by our speaker via the contact information that you provided at the time of the registration. So before we go, I would like to thank all the audience for joining us today and for the great questions that you raised during the presentation. And I would like to also remind you that there is a survey form with a few short questions. Please kindly take 1 to 2 minutes to respond. And thanks, Labroots. Lastly, this webcast can be viewed on demand, unlimited time. So Labroots will alert you via email when it's available for replay. We encourage you to also share that email with your colleagues or your friends who may have missed today's live event, but that are interested in this content. So until next time, goodbye and thank you.